So remember that plasma makes up 55% of the blood volume, and plasma is the liquid part of the blood. So plasma is going to be made up mostly of water, so that's really important to remember. And within the plasma, there are dissolved solutes. And the most numerous of the dissolved solutes would be proteins. So that would be important to remember as well. And also dissolved in the plasma, we have some electrolytes, so things like sodium ions, chloride ions, potassium ions, and other ions. And we also have nutrients, things like glucose, amino acids, and other things. We also have waste products, things like urea, which remember that comes from breaking down amino acids. We also have creatinine, which that comes from creatine, which comes from the skeletal muscle, and remember that's used to make ATP. We also have gases like oxygen and carbon dioxide and hormones, and so those will all be dissolved in the plasma. And if we take a look at the plasma itself and compare that to the interstitial fluid, which is the fluid that surrounds the cells and the tissues, the concentrations of all of these solutes between the interstitial fluid and in the interstitial fluid around the cells will be very similar except for the plasma proteins. So the plasma proteins will stay inside the blood in the capillary, but they really don't move out here into the interstitial fluid. And the reason that is because remember that the holes in the capillary walls are too small for the plasma proteins to fit through. And so those plasma proteins just remain inside the blood, and so they don't move out into the tissues. So that's why the plasma and the interstitial fluid are going to be different in their protein concentrations. This picture here just shows the composition of the plasma. Just reminding you that it's 55% of the blood volume, and plasma is going to be mostly made out of water. And out of the dissolved solutes that are in the plasma, the biggest solute that's going to be dissolved in the plasma would be the plasma proteins. And then we've got some of these other solutes listed here as well. Okay, that are also dissolved in the plasma. The plasma proteins have a lot of different functions. One of their important functions is they create osmotic pressure in the blood. I remember we talked about that before when we talked about exchange at the capillaries. So remember in the capillaries, we had osmotic pressure from the proteins in the blood. And remember that's going to attract water by osmosis because water likes to follow the solutes. So are these plasma proteins going to favor filtration or absorption in the capillary? So hopefully you said that it's going to favor absorption. And remember that's really important at the venule end of the capillary. That's what's going to pull the fluid back into the blood in the capillary at the venule end and return the blood back to the heart. So plasma proteins also will increase the blood viscosity or thickness. And how does this affect the resistance to blood flow? So remember this would increase the resistance and that would decrease the blood flow. So if you have too little of the plasma proteins, that will actually decrease resistance and increase the blood flow. So you've got to have the right amount of proteins in the blood to make sure the resistance is at the right level. So some of our plasma proteins will also help buffer the blood pH. And remember that buffers are like sponges, so they help keep the blood pH in the normal range, and if there's too many hydrogen ions, they can soak them up and remove them, or if there's not enough hydrogen ions, you can squeeze the buffer sponge and release hydrogen ions. Some of our plasma proteins also act as carrier proteins in the blood. And are they going to be transporting things that are hydrophilic or hydrophobic? in the blood. So hopefully you remember that things that are hydrophobic don't like to mix with all the water in the plasma of the blood. And so they will be attached to your carrier protein in order to be transported around the blood. So if you had like a hydrophobic hormone, like a steroid hormone, it would be attached to your carrier protein while it's in the blood. Some of our plasma proteins are used for blood clotting. So they're clotting factors, which we'll talk about at the end of the chapter. And then some of our plasma proteins are used for defense. They're actually antibodies. So we'll look at that next. And then we talked about in chapter 6 how some of our plasma proteins are actually going to be protein or peptide hormones. And then also some of our plasma proteins are actually enzymes. So if we look at our plasma proteins, there are 3 main groups of them. We have our albumins. These are the most common type of plasma protein. They have a lot of different functions in the blood. So they could act as carrier proteins, they contribute to the osmotic pressure of the blood, they affect the blood viscosity, and they do help buffer the pH of the blood. And then we have our globulins. So the globulins can act as carrier proteins, and they can also act as clotting factors. And then we have the gamma globulins, which are also called antibodies. And the antibodies are made by some of our white blood cells called B lymphocytes. And then the third group of plasma proteins, these are called fibrinogen. And fibrinogen are used in blood clotting, which we'll talk about at the end of the chapter. Okay, and then it's also important to know where these plasma proteins are being made. They're actually made by the liver. Okay, except for the gamma globulins or the antibodies, which are going to be made by some of the B lymphocytes. So if the patient has liver disease, they're not going to be able to make these plasma proteins very well. And because the plasma proteins have a lot of different jobs, as you can see here, they can run into a lot of different problems because of that. Okay, and then this is just from your book that just shows the components of plasma. So it's going to be made up mostly of water. 90% of the plasma is made up of water. And then the rest of it is going to be solutes that are dissolved in the water. And the biggest solute that is dissolved in the plasma would be the proteins. And then out of the proteins, the most common plasma protein would be the albumin. But we also have globulins and fibrinogen as well. And then we've also got waste products and nutrients. And then also we've got some ions and gases also dissolved in our plasma. You might have also heard of serum. So serum is different from plasma because it's going to be what forms after the blood clots. So here we've got a picture of blood that has clotted, and here's the blood clot on the bottom, and what's left here on the top is the serum. So the serum is going to be the plasma minus the fibrinogen and other clotting proteins. So that is what serum is. Okay, so down here we've got all the red blood cells, white blood cells, platelets, the fibrinogen and clotting factors that have all formed the clot down there. And then the serum would be the plasma minus the fibrinogen and the other clotting proteins. And so we would send the serum off to the lab to run tests on the patient, maybe checking for their liver function or kidney function. There's a bunch of tests we can run on the serum. And this picture actually is showing you a serum separator tube, and this kind of tan gel here, that is actually not part of the blood. That actually comes in the tube already, and it just separates the serum from the blood clot, and it just makes it easier to remove the serum without contaminating it with any of the blood clot components.